Well, good morning and welcome to the latest instalment of the National Sheep Association's webinar series. Over the last 6 weeks, we've been looking at a range of topics that affect sheep breeding and the sheep meat trade, in fact. And this week, it's the turn of gene editing. Gene editing has been thrust into the spotlight by DEFRA's announcement that they want to consult on the use of gene editing in livestock breeding. It's my understanding that DEFRA would like to change the description of a gene-edited animal from a GMO, that is genetically modified organism, to a simply gene-edited organism, to a non-GMO, and therefore legalize its use. We at NSA are holding this webinar, and we've invited two experts from the Roslin Institute who are involved in research regarding gene editing. We've invited those guys to inform us and put us into a position where we are able to respond to that consultation accordingly. It's worth saying, we are not here to sell the technology. We're not here to encourage people to use it. At the same time, we're not here to discourage people to use it. We just want to talk about the subject. We want to talk to a range of experts and put ourselves into a position where we know more about it. With that in mind, I would like to introduce two people we have on board from the Roslin Institute, Dr. Emily Clark, and I think I'm right in saying Dr. Simon Lillico, two researchers who have an interest in gene editing. Simon has an interest in gene editing and has several publications under his belt. And Emily has an interest in identifying gene editing targets. It is something that's going to probably take hours and hours to discuss thoroughly. So with that in mind, we just have an hour. I would like to move straight into the presentations. We're going to have two short presentations, then we will move into a panel debate with our other experts, and then I'll feed in question and answers from the audience as well. So we'll move straight to Simon. As I said, Simon is a research scientist. I'll get it right this time, at the Roslin Institute, and specifically has an interest in gene editing in livestock. So over to you, Simon, and if you could educate us, please.
I will do my very best. Okay, so where's my presentation? There we go, and share that. So yeah, I am a research scientist at Roslin. I have a long-standing interest in editing a variety of livestock species, either to make better models of human disease or to make disease-resistant or resilient livestock of relevance to the farming sector. So for my talk today, I'm going to give a short overview of the genetics underlying livestock agriculture. I'm going to start before recorded history, making a brief pit stop in the 18th century before moving on to the current state of play. I'll touch on the nature of genetic modification, or GM, and then explain how gene editing is different and give an example of how we can use it to benefit all of the parties concerned in the process. So modern humans have been around for about 300,000 years. And for most of that period, our ancestors lived in the landscape as hunter-gatherers. Our earliest domestication is thought to have been of the wolf around 10,000 years ago. And as with subsequent domestication events, the initial selection is likely to have been for tractable individuals, i.e. those that were least dangerous to our forebears and were docile enough that they could be effectively bred. Of course, in more recent years, selection of the dog has taken a sometimes bizarre turn, but possibly no more strange than some of the agricultural breeds one might encounter today. Jumping out of prehistory and landing in the 18th century, we have Robert Bakewell, and he is considered by some to be the father of the British agricultural revolution, because Bakewell suggested that breed improvement could be achieved if one were to select and breed from animals with desirable characteristics. And as a very brief aside, this thinking was formative in subsequent theories of a certain Mr. Charles Darwin. So as a result of this process of selected breeding for observed traits we find desirable to us, we have myriad livestock breeds that differ from each other significantly in both form and function. And selective breeding is not only the driver behind breed diversity, it's also the driver behind the spectacular productivity gains observed in Western agriculture, and that's exemplified in this graph of the milk yield gains in dairy cows in the UK herd over the last 25 years, during which time the size of the national herd has reduced, but the amount of milk produced by each animal within that herd has increased, so that we now produce more milk with fewer animals. So selection for the best animals over multiple generations is a hugely powerful tool to improve your trait of interest. But this approach does have limitations, and the major limitation is that you're limited by the genetic pool of the population from which you're going to select. In essence, if your trait of interest isn't encoded in the genome of your breeding population, you can't select for it. And genome editing, which I'm going to talk about, has the potential to offer an effective solution to this problem. But before I get on to genome editing, I thought it best to just give you a very brief overview of the genome itself. So you probably all remember from your days at school, but as a recap, most animal and plant cells contain a nucleus, and within that nucleus is the DNA. And this DNA is in essence an instruction manual for how to build an organism. So there are thousands of genes, and each of these tells the cell how, where, and when to make a specific protein. And all of these proteins have slightly different functions. So small changes in the DNA sequence can result in small changes in how, when, and where a protein is made. And this in turn impacts on how an organism is built and how it works. Genetic modification, or GM, is a blunt tool that allows scientists to take a whole gene from one organism, such as, for example, the green fluorescent protein that you find in some jellyfish, and you can insert that gene into the DNA of another organism, such as a mouse or a chicken or a pig. And as a basic research tool, this approach has been hugely successful, but despite some good potential, it hasn't really made much impact on livestock agriculture, due in part at least to adverse public perception of the technology. Gene editing is different because it doesn't involve moving genes from one species to another. Just as editing is the act of rewriting text, genome editing is the act of rewriting the existing genome. There are a number of different genome editors available to us, and you don't need to understand the specifics of how they work for the purposes of today. They are in effect all tools, and you can think of them as DNA scissors that allow us to cut the genome at a precise position and then rewrite the DNA sequence at that position. By making small changes to the DNA, we can alter how, where, and when the animal's own proteins are made, and thereby make changes to the form and function of the animal. So, as an example, I'm going to discuss briefly a viral disease of pigs that I've worked on called porcine reproductive and respiratory syndrome, which causes late-stage abortion in sows, death of young piglets, and a failure to thrive in older animals. There are vaccines against PRRS, but they don't cross-protect against the different strains of the virus, and in the EU alone, this disease is estimated to cost around about 1.5 billion euros every year. So this is a big deal in the pig sector. Before describing how I've used gene editing in an attempt to tackle this disease, I first want to show you this cartoon of the surface of a cell. So all of the cells in your body have a cell membrane around the outside of them, and embedded in that membrane a variety of proteins and fats and sugars. And these allow the cell to interact with other cells around it, and more broadly in the context of your body as a whole. The surface of a virus similarly has proteins on the surface, and these allow the virus to interact with your cells because they need to get into your cell in order to copy themselves. Now, Just to contextualize this, you're probably familiar with the spike protein of COVID-19 because you will have heard about it in the news. This is the protein on which all of the vaccines that we have are based. And the COVID-19 virus uses its spike protein to interact with a specific human protein called ACE2. Similarly, the porcine reproductive and respiratory syndrome virus uses proteins on its surface to interact with a specific part of a pig protein called CD163 that's found on the surface of pig immune cells. So what we did was we used genome editors to snip out a small section of the pig gene that codes for the pig CD163, removing the sequence that codes for the bit of the protein that the virus would normally bind to. And we generated a small cohort of pigs, and you can see here the 8 animals we used for our study. So 4 of these animals are controls, and 4 have the modified CD163 gene. And we challenged these pigs with the PRRS virus and showed that they were, in fact, as we had hoped, completely resistant to virus infection. So at the side of the graph, we have the amount of virus in the blood of the animals during the challenge study. And along the bottom, we have the number of days after they were infected. And the orange lines are the 4 control animals. So you can see the virus gradually goes up in these animals as they get sicker. The green lines are our gene-edited pigs that are completely resistant to the virus. And this is a big deal because not only can this approach be used to reduce the potential for animal suffering, there's also the added benefit to the farmer of reduced costs associated with the disease because their animals don't get sick, they don't die, they don't end up in landfill. However, perhaps the biggest plus is the potential benefit to society. So PRRS infections are the single biggest contributor to the use of antimicrobials in pig production, because when pigs get sick with PRRS, they also tend to get secondary bacterial infections. By reducing the risk associated with PRRS, we can reduce antimicrobial use in the pig sector, and therefore we can reduce the risk of antimicrobial resistance developing, and in turn, therefore, reduce the risk to human health. And I'd like to just finish with this thought. By selecting for observable traits, humans have been indirectly modifying the genomes of our livestock for thousands of years. Genome editors are just the newest tool available to us to do that, but they are just that, they're tools. Because of the huge potential associated with them, there is considerable resource being expended on their design and their redesign, and this evolution of the tool set is likely to continue for the foreseeable future. I think it's also important to understand that, as with any tools, genome editors can be used in a number of different ways, and I've only given you one example of that. There are, in fact, a spectrum of potential modifications that can be introduced into the genome using this tool set, ranging from alteration of a single DNA letter through to the targeted insertion of a foreign gene, so GM. You can use genome editors to deliver GM, but you can also use genome editors to edit. And determination of where edited morphs into GM is going to be a really important decision for regulators to make as the national debate goes on in this subject. And that is all I was going to say for now, and I'm happy to take questions later on. Thank you very much for your attention.
Well, thank you very much, Simon, for that brief but really informative presentation. It outlines perfectly the power of a technology like gene editing, and we'll come around to the questions later. But you know, it's amazing that you can do something quite as simple as that without any other side effects, removing a part of a gene that simply had an effect on that particular disease. And I'm assuming you're going to continue to monitor these pigs, and your research will go further. But it absolutely outstanding results to get a complete protection over that infection. So I'm sure there will be some questions on that front going forwards. But we'll move straight into Emily's presentation, and then we'll, as I said before, we'll move into the panel debate afterwards. So hopefully people will be scribbling down their questions. And over to you, Emily.
Okay, great. Thanks, Thomas. So just going to share my screen. Yeah, so Simon's given you a really nice overview of genome editing. So my job now is to talk to you a bit about how genome editing could be applied to the sheep sector. So genome editing can be applied to sheep in a number of ways that are described in this diagram. We don't need to think much about the ones in pink, orange and yellow today. They're in a different context. But as Simon mentioned in his talk, genome editing can be used to produce disease-resistant animals. In sheep, this would help improve welfare, productivity and sustainability because healthy sheep require less resources and are generally happier, I guess. So genome editing can also be used to investigate the function of genes, which is the work that I do in my research group at Roslin. So we use gene editing to understand, for example, muscle growth in sheep. So we can make small changes to the genome of cells in culture and look at how that affects their characteristics, and then think about how that might affect the characteristics of the sheep themselves. So then the main example I'm going to give you today is how genome editing can be used to produce genetically superior animals. And the example I'm going to talk about is called surrogate sires. So you might have seen surrogate sires reported in the media at the end of last summer. So the BBC and Farmers Weekly and many other publications ran articles about surrogate sires, indicating that scientists had produced gene-edited animals that could serve as superdads, and mostly publicized work that was done in goats at the end of last year. But this technology can actually be is also available in pigs and pigs and mice at the moment. So I'm just going to explain on the next slide what we mean by a surrogate sire. So surrogate sires are animals that have had their germline ablated. So what we mean by that is a gene called nanos2 is expressed in spermatogonial stem cells. The function of nanos2, this gene, is to maintain the spermatogonial stem cell population. So if we edit out nanos2, this should result in animals that have intact testes but lack spermatogonial stem cells. So basically that means these animals don't produce their own sperm, but they have functioning testes. And these animals are called surrogate sires. And we call them surrogates because they're then able to disseminate the sperm of elite sires. So what we do is culture the spermatogonial stem cells of elite sires and transplant them into these surrogate sires. These surrogate sires can then, instead of disseminating their own sperm or genetics, disseminate the sperm or genetics of the elite animal. Okay. So this is how surrogate sires work in terms of breeding. So the elite sire, we harvest spermatogonial stem cells from him, culture them, and then we transplant them into the surrogate sire that has the functioning testes. He then disseminates the sperm of the elite sire. He mates with an elite ewe, and then their offspring have the genetics of the elite sire and no genetics from the surrogate sire, because all he is just a carrier. So the advantages of surrogate sires are that the surrogate rams carry the sperm of the elite ewes. Surrogate rams carry the sperm of the elite sires. Instead of having one elite ram that can do only a limited number of coverings of ewes, we'd be able to have thousands of surrogate sires that can disseminate his genetics, making the reach of his genetics much, much greater. This provides a transformative step change in the way that the sector could disseminate the genetics of elite animals. And it would contribute to healthy and sustainable production through the dissemination of desired characteristics in these elite animals. So, so Mike Coffey, who's on the panel, and I have been developing this model for how surrogate sires could be deployed in the UK sheep sector. So the model is based on using genomic breeding values from genetic markers and the development of stem cell technology. to identify elite animals and then provide their sperm to multiplier units that would be populated by surrogate rams. These surrogate rams would have all the characteristics of a good ram, so they'd be like medium size, they'd have good libido, and they would also be vaccinated and managed very carefully, so that they would then disseminate the genetics of the elite lambs, the elite rams, through lots of lamb finishing units that would be distributed across the UK. These lamb finishing units would be from hill to lowland, and they'd be distributed at national and regional scales. So we fitted a sort of feedback loop into this model, where information on breeding priorities in these lamb supply units, such as feed efficiency and health traits, could then be fed back to the breeding nucleus. This kind of approach would also help to minimize inbreeding from this few number of animals in the breeding nucleus, again by measuring inbreeding in these supply units. So we think this model is a one potential model, and at the moment we're looking at potentially doing gene flow analysis and cost benefit analysis to see how it would work in practice. Okay. So I should say that we're probably still 5 to 10 years away from deploying surrogate sire technology in the sector. This technology is definitely not going to be available tomorrow, and the model I've shown is some way off. That's not to say that kind of a model couldn't be deployed for other existing technologies such as AI. So it's important, I think, given how fast the technology is moving, that we have the infrastructure in place when the technology is ready. So we have now tools as well as technology to identify genes at a whole genome-wide scale that are in driving characteristics, where it took us sometimes 15 years to identify a gene that was driving a characteristic in sheep. We can now do that much more quickly with very advanced technology, looking at from a single gene up to the whole genome. We're working at Roslin with governing bodies and regulators on the dialogue around genome editing. And this is really important, particularly recently with the consultation that's been launched by DEFRA. And we've worked with other groups, including the recent Nuffield report on genome editing in livestock. So currently, genome editing is very tightly restricted under EU law. And as I just mentioned, the UK government has launched a consultation on genome editing to modify livestock and food crops in England. So we do need support from the sector for this research, and also support just generally feeding back to DEFRA, and we're very keen to engage as much as possible with the sector, especially around the discussion that's ongoing at the moment. And yep, Simon and I and Mike are very happy to answer questions. Thank you for listening.
Thank you very much, Emily. One thing that it just sprang to my mind just there, where we were talking about the surrogate sires, I read the consultation document last night, and it is to change the status of genetically edited animals that could have been produced by natural means. Is that an obstacle to the surrogate sire idea, or is it something you think could fit into that description for you, Emily?
So, okay, yes. So my understanding of surrogate sires would fit into that description. So we're just, in this case of surrogate sires, we're removing the function of a gene, and the surrogate animals would be edited. The elite genetics that we disseminate through the sheep population is not edited genetics. So they would represent a model where we're not introducing any kind of different DNA when we're removing something, and we're not necessarily disseminating that further. So,
and we see that as something that's important, that edited animals don't enter the food chain.
So yeah, so that is a complicated question, and it's related to two things. So as Simon mentioned in his talk, you can there are kind of two ways. There's removing a gene, and there's inserting a new piece of DNA that's different, and it depends whether that change is already is already present within a population of animals or a species. If you're doing something that's not already present, that's very different to doing something that's already naturally occurring. And the regulation is likely, I suppose, to be different around those things. So if we're doing something that's already there, and that is referred to as a naturally occurring change, that's much more acceptable, I suppose, than if it would enter the food chain than if it was something we'd inserted into the DNA and then that entered the food chain. That's quite a different discussion. So these are quite different things, and will be regulated, I think, differently. I don't know if Simon has anything to add on that point.
Yes, so it is, as you say, it's a complicated question, and how the regulation will work, we're still very unclear. Mm-hmm. It is quite possible. So we don't have a full genotype for every sheep and every pig and every cow on the planet. It is quite possible that you can find, if you were to look at every genotype, an animal with a non-functioning nanos gene, for example, or an animal that happens to miss the bit of the protein in CD163 that makes the animals resistant to PRRS. The difficulty is, so these are small genetic changes, and small genetic changes happen all the time. Every time mummy and daddy love each other very much and you get offspring coming out, those offspring have somewhere between 50 and 100 genetic changes that neither of the parents have, because the genome rearranges during fertilization. So small changes are likely to be out there. So an argument could therefore be made that any small change could, in theory, be produced by natural breeding.
I suppose it just depends on the interpretation of the rules, doesn't it? Whether or not you'd prove that it had already occurred naturally or not.
If it has already occurred naturally and has been observed and documented, that's easy and clear cut. We're taking something and moving it from one breed to another breed without the baggage carryover that you would get from a standard breeding approach. We can just take that desirable trait and move it.
Okay, I think I had Mike's hand up there to contribute. I'll tell you what, I'll just introduce the panel who are going to now feed into the debate. We have William Hairsign, who is a retired professor of agriculture with a long-standing research interest in sheep breeding and sheep reproduction. We have Amanda Carson, who is the Sheep Vet Society president since September of last year. And we have Professor Mike Coffey, who you've already heard works with Emily, and Mike is a professor of livestock information, breeding and genomics. All three are members of the FANGA committee. That is the Farm Animal Genetics Resources Committee, is that?
Yeah.
And right, and I'll before I get well and truly out of my depth, I'll hand over to Mike, because you had your hand up there, Mike. You were trying to feed into that discussion.
Yeah, I just wanted to simply make the point that I'm guessing that breeders have naturally produced infertile rams in the past, but they've never made it into the population for obvious reasons. And gene editing is therefore a mechanism of replicating what many breeders have already done in practice.
Right, that's interesting. It is interesting that fine line that we are treading there to try and make sure that the description fits one way or the other. Going forward into the discussion, I would like to raise some considerations that we at the Sheep Association would like to clarify. And I suppose one of the main considerations is, how does the gene editing differ from genetic modification, and why should we class it as something completely different to the existing technologies? We would like to know the benefits and risks to GE. I think we've heard some of the benefits clear in Simon and Emily's presentations. What risks does it pose to the sector? Is it something that could be perceived as Frankenstein science? And how well do you think it'll go down with the public? And I think most importantly, from my perspective, is what does gene editing offer over and above what we have at the moment in terms of single gene selection and things like that are actually being used at the moment. We'll probably try and get to safeguarding and tech governance towards the end of the discussion if we have time. But if we could, given those 3 points that I just highlighted, Simon, how does it, if you had to give a brief description to a member of the public, what would you say was the main difference between gene editing and GMO organisms?
So GMO, as I said, has historically been the moving of a gene from one species to another. So you can take whatever gene you're interested in from whatever species you're interested in and put it into another one. And so this is, as a scientific tool, it's been really useful. It's also been used for producing livestock, for example, that make pharmaceutical proteins either in their blood or in their milk. But it hasn't made it to the table largely because it's had the Frankenstein label associated with it. So there are some real potential benefits that could be derived from GM being part of livestock agriculture, but I don't see us going down that route at the moment. And I'm not trying to sell it. It's, that is what it is. Gene editing can be used to produce GM animals, right? We can use to improve the efficiency, but gene editors are just tools. They allow us to break DNA at a very specific place in the genome. We can design the tools to do that, and then we can reform the DNA at that position. So we can make very precise, small changes. So, for example, there is a gene called myostatin, and myostatin is a negative regulator of muscle growth. What that means is the more myostatin you have, the smaller your muscles are, and the less myostatin you have, the bigger your muscles are. So Belgian blue cats, for example, have a mutation in their myostatin gene, which means that they don't make the myostatin protein, and they get really big muscles. Because it's relatively simple with gene editing to make a gene non-functional, if that's what you're choosing to do, you can use a gene editor in any species to knock out the myostatin gene and make a big muscly animal. And so there's a company has done that in tilapia, for example, in South America, to make tilapia that get to market value, to market size a lot faster because they grow faster. And the South American regulators have ruled that this is not GM, that they're not going to regulate it. It is something that could happen naturally. It's not something that has been observed in nature in tilapia, but it has been observed in nature in other animals.
In reference to myostatin in particular, there are several different variants of myostatin. The myostatin gene, aren't there? It's clear that some animals that possess myostatin mutations don't necessarily grow as fast, although their muscling is more pronounced. Are there variants out there that don't grow fast, or some that are extremely muscled and grow fast?
Yeah.
Is there something that we know a lot about at the moment?
It's something that we know quite a bit about. So if you take the tech cell, for example, tech cell has a mutation, but the mutation is not in the myostatin gene itself. It's in a gene that regulates the expression of the myostatin gene. So they still have myostatin, but they have less of it. So they grow bigger. I think Calipiche sheep also have a mutation that influences myostatin expression. So the simple answer is yes. I have to say I'm not a myostatin expert per se. I think what I've told you is everything that I know about myostatin. But there are subtleties to it. You don't have to knock out the whole gene. In any of these things, we can also make changes to the genome that influence how much of a protein is made, where the protein is made, when the protein is made, that will influence form and function of the animal.
It's just interesting. Last night I read a very short piece. I couldn't say how accurate or inaccurate it was. That Highland cattle also have the same myostatin mutation as Belgian blues. It's just not, obviously the regulation is different in Highland cattle to Belgian blues.
I'd say that's not one that I was aware of. So
I think William was nodding there. Could I bring you in there, William?
Well, I'm no expert in it, but like you, I've seen the information which says that they carry the same mutation, but it doesn't express. And yeah, in quite a number of instances, we have a situation where a particular biological function is not regulated just by one gene, but by a number of genes that interact. So we need to be careful in making an interpretation. Can I just go back onto the GMO gene editing side of things? If I turn the clock back about 30 years, one of the first, in those days, GMOs that was produced was the flavor saver tomato. And the benefit of the flavor saver tomato was it had the gene that produces the enzyme that causes softening of the very ripe fruit inactivated. So that you could leave the tomato on the tomato plant until it got fully ripe, so it's got more flavor, it's actually a sweeter type of thing, but it doesn't then sort of disintegrate and go very soft and squishy on a supermarket shelf. But that's all got wrapped up in the GMO side of things. And I think if we go back to those days, there was, how shall I put it? I think scientists didn't engage in dialogue with the general public in a way that was helpful. The view was they won't understand, there's no point in telling them about it. And then, of course, the whole thing collapsed and we got into regulation which says no GMO. We need to be very, very careful on this side of things with gene editing. I don't want to sound negative, but I think we need to take the public with us. And I think, you know, the concept that you can actually produce animals that are naturally resistant to diseases that cause a real challenge in the production environment. You reduce antibiotic use, you therefore reduce the likelihood of antibiotic resistance developing that can have consequences in the human population. There are some good news stories that we can use, but we need to be very careful in using it.
Yeah, it strikes that what you're saying there, William, is that, you know, if we pursued a route that would improve the health and welfare of our animals, we're much more likely to get the public on side than if we did something like fetched in myostatin mutations to make double-muscled animals for the profit of farmers. To start with, there are many examples of the health and welfare of our animals, and I think Simon's research showed that very neatly. regulation going forwards is going to be key. But if we'll move on to that in a bit, we'll just try and cover the technical basis for the moment, if that's okay. So what, if we can cover this for the next few minutes, I think the big question on most people's lips is that what does gene editing offer over and above what we have at the moment? If you could quantify how much more powerful, or in your mind it is or isn't, than the single marker technology. One thing that I worry about with single marker technology is the genetic linkage. You're not necessarily always just selecting what you're looking at, you're selecting what's attached to that gene close by and on the same chromosome, if you like. And if you're not looking at the relevant phenotypes, you can easily breed something in that you're not looking for. That's my issue with single gene marker technology. I'm not here to really give my opinion, but if you could outline how gene editing offers a different route to that. I think either Simon or Emily, if you'd like to come in there. Go, Simon.
Okay, so with a standard breeding approach, let's take the myostatin as an example. I'm not pushing myostatin at all, but as an example, let's say you've got your Texel sheep, which are great for meat. And so bear in mind, I'm not a sheep expert, but you've got another breed of sheep, which is excellent for wool, and you want your woolly sheep to be more meaty. So you could cross your Texel and your beautiful woolly sheep, and you'll end up with some hybrid in between the two, which you then need to select and cross over many generations to get a meaty woolly sheep. Using gene editing, because we know the genetics that underlie the Texel meatiness, we could introduce that directly into the genome of your woolly sheep. So in one generation, you go from a skinny woolly sheep to a meaty woolly sheep. So in essence, that's one of the real powers of this technology. You can introduce a single trait of interest without the genetic baggage that you were just talking about that occurs in a standard crossing program.
Yes, Mike.
Sorry, yeah. I just wanted to make the point that the conversation obviously is kind of wide ranging, but the specific point of gene editing in the model that Emily proposed was to knock out one gene to create infertile rams that could then be the distribution method for sperm from other rams. Now, those rams don't need to be gene edited. They can be elite rams that have been selected conventionally within a nucleus breeding program to have very desirable traits. And the selection within that breeding program can be very intense and fast to create these super rams. And the only point of gene editing in that scenario is to make the surrogate sires infertile so that they can carry those sperms. So we're not talking about disseminating thousands of animals whose offspring have been gene edited in that example. Clearly, Simon has already mentioned there are many examples where we might be able to change sheep so that they're more desirable for specific characteristics. But in the scenario that Emily was painting, the use of gene editing was simply to stop a ram producing his own sperm.
And am I right, Emily? There are safeguards in the approach that you are suggesting to monitor the effect of inbreeding.
Yeah, so I think actually Mike's probably better to answer that one, but yeah.
Yeah, again, I think I've answered it in the Q&A thing that I mean, inbreeding is an issue in small closed flocks. It's an issue in the dairy industry where many of the animals are offspring of related sires, but all of it is manageable. if you have information on the genome of the animals that are participant in the breeding program, it is very manageable. So I would say that inbreeding in sheep is much less of an issue than the benefits that such a technology could bring. You know, you wouldn't say, let's not deploy this technology because we may in 50 years time end up with some inbred animals. There are already inbred animals, and farmers manage that locally. It's as easy to manage inbreeding in a very well recorded. Myself and Will have spent a lifetime trying to encourage farmers to record things, and one of the things that you can benefit recording with is to prevent inbreeding or control it.
I think one of the great assets of the UK sheep industry is that we have a variety of sheep breeds, and that we, you know, it's proving itself in that we fill a lot of different niche markets with all our different products. One thing we have is a stratified sheep system that it's not the whole industry, but it's a significant part of it. In the stratified system, there's clearly there's an emphasis on hybrid vigor and what that delivers for maternal traits and complex traits that are controlled by many different genes rather than one or two genes. Do you see gene editing have an effect on the stratified system, and how would it fit into that system? It might be a bit of a woolly question.
Yeah. Yeah, it is a woolly question. Yeah, but I think the point there Emily made is that this is a technology that is currently being considered for its place in the legislative framework. And the important thing is to make sure that it goes into the appropriate legislative framework such that the benefits it brings can be deployed, my expectation to be over the next 50 years. So it's not going to change the UK stratified sheep industry next year or in the next 5 years or even the next 10 years. But when the technology does become available, in each of those stratifications, you can amplify the genetic superiority of that stratified sector, be it the maternal side, be it the growth side or the woolly side or any other side. And Will's far more of an expert on stratification than I am, but stratification itself is a mechanism. And if each layer was genetically far superior, the overall system would work better.
That's a good description. Thank you very much, Mike. I noticed, Amanda, you've been quiet so far. From the Vet Society perspective, what do you make of gene editing?
Well, I have to say that I'm in awe of all the scientists that work at Roslin, because this is such a complicated topic, and I don't really understand it, to be honest, because genes are incredibly complicated, and it would be great to say there's one gene that we can put into all sheep that will make them resistant to gastrointestinal parasites. Wouldn't that be marvelous? But it doesn't work that way. You know, genes are switched on by other genes and switched off by other genes. There's lots of interrelationships, but it's an area that we're still exploring. And as Simon alluded to, there is the potential with this technology to look at specific diseases and perhaps identify particular genes that might help us to control diseases better. And I think that's hugely important. And it's something that we, you know, really need to get on top of, as well as, you know, what benefits it can bring to the sheep sector in improving production. But production also is related to the health of animals. And I see a lot of these technologies as bringing huge potential benefits to the health of animals. I mean, look at some of these diseases like OPA. We don't have a test for OPA in the live animal. We don't have a vaccine that we can control it with. But there's likely to be something that we can potentially find with animals with this disease through all of this research. So I think it's hugely important, incredibly exciting, and well done to all the people at Roslin and other scientists.
Thank you very much, Amanda. One panelist that isn't with us today, I think I'm right, Ian McDougall. He sent some questions, some views over. If I could just outline quickly, I'll summarise Ian's question and put it to the panel. Ian said, while these technologies are interesting, I think they are unlikely to gain much traction within the British sheep industry for some time. We have created an image of lambs being green, well cared for and natural, not manipulated by humans simply for profit. There is the risk of the public who are already being persuaded away from lamb by veganuary etc. to be further driven away by the Frankenstein syndrome, or providing the vegan movements with new reasons to bash sheep farmers and promote the non-eating of lamb. The general public are likely to see gene editing as messing with life. Discuss.
Don't forget, in the context of animal and plants, gene editing can apply to plants just as it can to animals. And, you know, following on from previous comments, you imagine if you could breed plants that were drought resistant or flood resistant, or, you know, resistant to some of the diseases. I saw a tweet last night on a farmer tweeted 40 hectares of cabbage destroyed by some kind of cabbage eating weevil of some sort. You know, clearly such thing. That's why farmers farm. You know, they work with the wildlife, they work with the landscape, they work with the soil, and all this technology is doing is allowing them to do that job much better. And it applies to plants and animals. It's just the same. You know, these are not Frankenstein animals. And I refer you back again to the point I made earlier, that in the context that Emily spoke, we were talking about using gene editing to simply allow us to create animals that are the delivery mechanism of potentially unedited sires, rams. So conventionally bred rams can have their semen distributed far more widely, and the benefits of those rams being made available to far more farmers through the use of a single gene edit in the carriers. Nobody is suggesting you would eat the carriers. I don't quite know what happens to an old ram once they've serviced hundreds, or in this case, thousands of sheep.
You eat them.
But, yeah, you eat them. Okay, fair dues. Well, maybe that's what the legislation is for, to appropriately allow the technology to be used in a way that's acceptable to the consumer, acceptable to society and government legislators.
Mike, would it be fair to say that proposal is almost like an alternative to artificial insemination?
Yes, that's right. I mean, I wrote down here that in the dairy world, the AI technician is the surrogate sire. The AI technician is driving around with a van full of semen in the back, and he gets out at each farm, or she gets out at each farm, and inseminates thousands of cows. That's all that this is. Instead of it being a van, it's a ram. That's a good line, isn't it? You can. Yeah. We'll have to trademark that and use it. So instead of it being a man with a van, it's a ram with someone else's sperm.
It's an interesting lead into the regulation. You talk about crops that are drought resistant and things like that. There's obviously the really famous example of the golden rice being limbo for decades, where it could have really helped lots and lots of people. That's an example of GM specifically, isn't it? Do you see a situation where it was developed by a large tech company? Do you see the possibility of large tech companies having a big influence on the way things are going to be in the future and the way this is sold to farmers, if you like? Or will it be something much more grassroots, where you could take your sheep to the AI center and the AI technicians would be able to do an embryo flush and gene edit?
I think that very much, Thomas, depends on the sheep industry. Because the drivers for change are massive. I mean, you know, the planet's dying in front of us. So the desire to reduce greenhouse gas emissions is immense. And of course, it won't take many large global companies to realize the potential for doing this sort of thing to become involved. And that's why I've been a very keen advocate. Myself and Emily have been working very closely with individual sheep farmers to work out ways of being able to get the framework in place such that these technologies can roll out as and when they need to improve sheep, to enable the sheep industry to regain or retain control of its own destiny. You're absolutely right. There are very many large multinational companies sitting at the sideline waiting to influence what we eat in the future. And I draw your attention to the vegan movement, which is highly politicized and controlled by very large international food processing companies. The likes of Nestle and others have a very keen interest in the population eating ultra processed foods. And I'd much rather put my faith in tens of thousands or millions of global farmers to keep us safe than I would half a dozen global corporates.
Is there a risk then that, I'll feed a question in from the Q&A, is there a risk that elite or surrogate animals could be patented, so controlling their access?
Well, again, yes. If a company controls the process like Genus does with its pig, its pig lines, and if the market wants to buy Genus pig lines, they buy them from there. If they don't like Genus pig lines, they buy someone else's pigs. Genus actually is a British farmer-owned company. It's owned by thousands of ex-dairy farmers. So there's no real reason why a good sheep breeding company shouldn't arise that is owned by UK farmers. I think, Will, there's already an example, isn't there? Innovis is a UK sheep breeding company.
Yeah.
There could be dozens of Innovises running around the country, focusing on individual market niches, different types of sheep, different markets, tying up with retailers to create value-added food chains, all sorts of possibilities. But the potential to do that is in the hands of farmers right now. And
so, if I could bring in Simon and Emily on that front, as researchers, you obviously do all the legwork. What would you? It seems to me like it would be undesirable from your point of view for large companies to take this forwards and take ownership over the technology.
Not necessarily. I'm not sure what Simon thinks, but we at Roslin, we have partnerships with some large companies for pigs and poultry, and that works well. Obviously, we rely on science funding to do the work that we do, and sometimes companies can bring, additional funding, can really help us answer additional questions. So it can be beneficial. If Simon has something to add.
So I agree. The pig that I discussed actually was co-funded by Genus, and certainly at this stage, having a big player like that on your side, they're interacting with the FDA in the States, for example, to try and get market acceptance to get these animals to the marketplace. So from my perspective, the involvement of big companies is often a beneficial thing.
Go on, William.
I think I'd just like to add a degree of realism into the discussion. When we look at the pig industry, we've got big breeding companies involved. When we look at the dairy sector, we've got big breeding companies involved. And you can imagine that there could be specific interests in gene editing under some circumstances. When I look at the sheep industry, I mean, for heaven's sake, what is the level of penetration of genetic performance recording and genetic improvement schemes in the UK sheep industry? And the answer is it's pitifully low.
Yep.
So I can't see individual sheep farmers having any interest in gene editing for the next 50 years. I think the second thing I would say is coming back to public acceptability. and regulation. It's not just what we think in the UK that's important. It's what the various markets that we supply products into actually believe. And I know that there is debate going on within the EU because there are some countries who believe that gene editing should be treated differently from GMO. But currently the EU position on this is that the same thing, and they don't want it.
So if I could just feed one more question in from the Q&A, which is related to what William's saying there. Regardless of what happens with the consultation, whether or not we can roll this technology out, whether or not we want to roll this technology out to industry, is it, in your views from the Rosalind Institute, will your research be able to go ahead for years ahead, given that they may still be regarded as GMOs? Because it's obviously it's one thing to use it in industry. It's quite another to keep the UK's research industry at the forefront of science and keep up with all these nations that are pushing ahead with this sort of thing. I think everybody would admit that you've really got to be in it to win it, and to identify any potential benefits from gene editing, we would certainly have to have a really strong research industry. Are you, Emily and Simon, confident that your research will be supported going forward?
Yeah.
Regardless of the outcome of the consultation?
I think so. With respect to the research funding, I would see that likely continuing. The advent of genome editors has made this a really good time to be a scientist. There is so much that we can do by way of scientific discovery with these new tools. From my own perspective, I'm obviously acutely interested in the outcome of the consultation, because I like to think that what I do is more broadly useful than just scientific discovery. I love talking about the PRRS pigs, because if we can get those pigs to market, then something that I've done has been really useful. And, you know, I like that. But
it's very hard to argue with making animals healthier, isn't it?
Absolutely. So I think anything that we can do in this space that improves welfare and improves the health of the great British and world public is good.
So I'm going to get into trouble for running over. Already I've run over a little bit. But with regarding your research, Simon, with the pigs, I know they're not sheep, but you know, genetically things work in a very, very similar way. It obviously shows huge promise. Going forwards, do you have a line of pigs that carry this genetic change, and are you going to monitor them for, because obviously the CD163 gene is involved in immune responses. Are you monitoring those pigs for potential side effects you haven't looked at yet, potential susceptibility to other diseases that are out there?
So we have a small cohort of pigs still here at the Institute, but we don't have money to keep them for much longer. But realistically, because we've done all of this with Genus, as far as we're aware, Genus they're remaking the pigs in their own elite animals, and they'll do all of that due diligence. And the bottom line is the market will decide whether these are useful animals or not. I think they are, but I'm biased. The farmers in the US and more broadly will decide whether these are animals that they want, and if they are more susceptible to other diseases. Don't get me wrong, we have no evidence that they are. I don't anticipate that they will be. But if they were to be more susceptible to another disease, for example, it would be up to the market to decide, well, we're willing to take that risk if it means they don't get PRRS, or we're not. So as with any new breed of animal or any new breed of plant, the market will determine the value and how it gets taken forward.
Emily, do you have anything to add there?
Before
we wrap up?
Yeah, so I guess our main funder is BBSRC and the UK Research Institute Collective, and they have a main theme called Biosciences for Health and understanding sustainability in agriculture. And I think funding for this research was one of their key priorities. So I would expect that the potential that we have to do research in this space is great, and then we're just really hoping that there'll be support going forward from all of the sectors that have an interest in this technology.
Good. Thank you very much, Emily. Thank you, Simon, as well, for offering your expertise. Thank you to William, Mike, and Amanda. I think it's been a really rewarding session. It's not going to go away. It doesn't matter what the outcome of the consultation is. There's clearly a desire there to push things forward from DEFRA, but hopefully we'll put ourselves into a position where we can answer the questions in the consultation with a clearer head about regarding what gene editing is. It's exciting times for anybody that's interested in genetics, and the future is certainly interesting. The sheep industry is in a strong place, and I'm sure that we'll all be talking again at some point, and this isn't a discussion that's going to go away. So I'll thank you very much again from myself and from everybody at the Sheep Association. Thank you to everybody that joined in the audience. Before I call an end to the meeting, could I just remind you that next week we have a webinar on, at the same time, 8:30 next Wednesday morning. We have a webinar on a farmer's guide to the halal sheep market, an ever-growing market in importance for UK sheep, so it's bound to be interesting. After that, because of the time of year, we're all very busy, lambing and whatnot, the webinars will move from a weekly slot to a monthly slot to the first Wednesday in every month. So tune in next week for the discussion on halal, and then thereafter every month. So again, thank you to everybody for tuning in. It's been really rewarding. I'll see you next time. Thank you.
Thank you. Bye-bye.
Bye.
Thanks, everyone.